Hey y'all, Miss Mayhew here, and we're going to go over the rest of Chapter 8. So far, you've already covered Chapter 8.1, that the genes are particulate and are inherited according to Mendel's laws. We're going to start here with Concept 8.2, that the alleles and genes interact to produce phenotypes. Now, the first thing I want to start off is explaining the words allele and gene to you. These are words that we use really often in this unit, and they might not have actually been explained to you before. So an allele is an option for a gene. And a gene is a section of DNA on a chromosome that codes for a protein. Now, when we're talking about these alleles, we refer to them in two different ways. So we can call them the wild type allele. This is the allele that's present in most of the population. The other alleles of that gene, so the other options for that gene, are mutant alleles. Okay? Other alleles or other options for genes arise through random mutations. Genes that have a wild type allele that is present less than 99% of the time is called polymorphic, meaning there's multiple options or multiple alleles. Typically, when we think of genes and we think of alleles, we think of genes that have two alleles. So two options, for example, freckles or no freckles. Okay, but sometimes there are genes that might have more than two alleles. We refer to these traits in these genes as multiple alleles, so meaning more than the two options we're used to seeing. An example could be the coat color in rabbits. So if we're looking at coat color in rabbits and we were thinking about a normal situation with the alleles, we would expect to see genotypes with capital C's or lowercase c's. But in this case, we have more than those two alleles. We also have this lowercase c with the superscript CHD, or the lowercase c with the superscript lowercase h. So there are multiple forms of the lowercase c allele. So this is a good example of multiple alleles. Another form of non-Mendelian inheritance is incomplete dominance. In this case, these alleles are neither dominant nor recessive to each other. So heterozygotes are going to have an intermediate phenotype. I usually remember incomplete is in between, or incomplete is intermediate. An example could be flower color in snapdragons. So as you see here, if we have two homozygous parents and they are crossed, we end up with an F1 generation that has 100% heterozygotes. Those heterozygotes are neither red nor white, but instead there's some intermediate or in between, pink in this case. Another form of non-Mendelian inheritance patterns are codominance. This is when you have two alleles of a gene that both are going to express the phenotype in the heterozygotes. Blood types are a great example of codominance and also multiple alleles. Okay, the reason we can use blood type as an option for multiple alleles is because, as you can see, we don't just have the two alleles. In this case, we have more than that. We have the A, the B, and the O. The reason this is an example of codominance is because A and B are codominant to each other, meaning if you have both of them are going to be expressed. I kind of think of codominance, makes me think of co-teachers or co-pilots. They're both going to be there. So in this table, we can see that there are A surface proteins, B surface proteins, or no surface proteins. When you have the AB group, that means that they're going to express both A and B surface proteins. Now, one important concept for you guys to understand is something called epistasis. Okay, what this means is that genes don't always act independently of each other. Sometimes the phenotypic expression of a gene is influenced by another separate gene. So one part of the chromosome that codes for a protein could be influenced by another piece of DNA somewhere else. So for example, if we're looking at allele B, so gene B, you have two alleles, capital B and lowercase b. black is dominant to brown. But there's another gene somewhere else we'll call gene E. Okay, this one has an allele or an option for that gene that's dominant to lowercase e. The allele E that's dominant is going to be for pigment deposition, so meaning that there will be pigment made. Okay, but the lowercase allele, the recessive allele, is that no pigment is going to be made. So gene E is actually going to determine the phenotypic expression of gene B, because if you inherit that lowercase allele, the recessive allele for E, that there's going to be no pigment made, it doesn't matter if you have the dominant or the recessive allele for B for the pigment if you're not going to express it. This is the case in labs. Okay, yellow labs are not going to express any of the pigment, either black or brown, because they have those two recessive alleles that code for no pigment. Okay, but if a lab inherits a dominant allele for the E, the expression of pigment, that's when the capital or lowercase b comes into play. One way genetics is used outside of biology and outside of strictly scientific research is in agriculture. So for example, there's a concept called hybrid vigor. Okay, this basically means that in certain cases, hybrid offspring are going to grow larger and produce more seeds than the parental varieties. So whenever these are crossed and combined, those alleles are combined, you end up with more productive crops. This is because now you have more genes interacting with each other. Okay, vigor, for example, is a complex trait. Okay, most of these complex phenotypes are determined by multiple genes. Okay, whenever there's multiple genes that affect one phenotype, in this case, like the productivity, okay, those are usually referred to as polygenic traits. So multiple genes affect that trait. Most of these are quantitative traits that can be measured rather than those that can be assessed qualitatively, although there are some that are both. Now, your genes themselves are not the only thing that are going to affect your phenotype. Okay, your genotype and the environment often interact with each other to determine the phenotype. Some examples are point restriction coat patterns, or coat patterns that are affected by a certain threshold or a certain point. For example, there are rabbits and cats that have a mutant allele for the coat color gene. Okay, this enzyme that is encoded by that gene is only going to work at temperatures below 35 degrees. So anything above that is going to be inactive. Okay, these extremities, so like their ears, the smaller, like their paws, the parts that are colder than the main body are going to be a different color because of the activity level of that enzyme being made. So when we're talking about the effect of the genes and the environment on the phenotype, there are some vocab words that we need to go over. Okay, the first one is penetrance. This is the proportion of individuals with a certain genotype that show the expected phenotype. So how much of the population that has the genes that code for this trait actually express that trait. Expressivity is going to be the degree to which the genotype is expressed in an individual. So how much of that expected trait is shown. These words are important in especially medical fields and medical research. So for example, many people, but not all, who inherit a mutant allele of the gene BRCA1, so the BRCA gene, develop breast cancer. This mutation is said to be incompletely penetrant, meaning that some people can inherit that gene, that allele of the gene, and not develop breast cancer. Okay, we can also use expressivity in the conversation around breast cancer. Okay, this mutant allele is also related to ovarian cancer. So there are some women that have this mutant allele for the BRCA gene that develop both breast and ovarian cancer. Okay, but some women with this allele could only develop breast cancer. So, because there are different variances in how this gene is expressed and how much of it is expressed, we can say it has variable expressivity. Now, when we're talking about genes, the sequence of DNA that's on a chromosome, these are located at a locus. This is essentially where on the chromosome that gene is. Now, these loci matter because of genetic linkage. Okay, this is where certain genes that are on a single chromosome can alter their inheritance pattern because they are linked. This genetic linkage was discovered by Thomas Hunt Morgan using fruit flies or Drosophila. Now, these flies have been used a lot in genetic research because of a few reasons. 1, they're small. 2, they're easy to breed. And 3, they have a short generation time. Now, he discovered this when he was doing crosses with the Drosophila and did not get the expected ratios according to the law of independent assortment. Instead, he noticed that some alleles for body color and wing shape were being inherited together. They weren't being shuffled like you would expect. His hypothesis that the two loci were linked on the same chromosome and could not assort independently. So he used the null hypothesis that the alleles are going to assort independently. And just like we would use in our actual AP exam or in AP biology, he did a chi-square analysis. He looked at how many he would expect to see, and he looked at how many he actually observed. Then he used that data to say that he rejected that null hypothesis. But here's the catch. The linkage wasn't absolute. Like, it wasn't that they never recombined. There were still some offspring that had a recombinant phenotype, resulting from that crossing over that we would expect to see in prophase 1. So you can see that there was crossing over that did occur. So we started to discover and started to look at recombination frequencies. This is the recombinant offspring that appears in a proportion. And here's the important part. The recombinant frequencies are higher for loci that are further apart on the chromosome. So the closer two things are together, or the closer two loci of two genes are to each other, the less likely it's going to recombine. The further apart they are, the bigger chance they have to recombine and cross over. So here is an example of some results that he got. Now, using these recombinant frequencies and how often these different genes assort independently from each other, you can use this information to infer the location of genes along the chromosome. Again, because the further apart they are, the more likely they are to recombine. The closer together they are, the less likely they are to recombine and separate. Another reason fruit flies are easy to use is because they have 1 pair of sex chromosomes and 3 pairs of autosomes. So they really only have 4 sets of chromosomes. Now, males have an X and a Y sex chromosome that are different in size, kind of like you would see in humans. And many genes on the X chromosome are not present on the Y. Thus, males only have 1 copy of some of the genes, so they only have 1 allele. Genes that are present on a single copy in a diploid organism, kind of like these, are called hemizygous, meaning there's only 1. Another important discovery made by Morgan's experiments is that it showed that the gene for eye color was carried on the X chromosome. The wild type, or the most oftenly seen type, is red, and the mutant allele is white, which is recessive. When he crossed these organisms, he noticed that the inheritance patterns didn't follow regular Mendelian genetics like he would have expected. When this happens, we refer to these as sex-linked inheritance patterns. Okay, this is when the inheritance of a gene is carried on a sex chromosome. In mammals, the X chromosome is larger and therefore carries more genes than the Y chromosome does. Most of the sex-linked inheritance patterns are going to be involving genes that are carried on that X chromosome. You can see examples of this in pedigrees especially. Now, some patterns to look for when you're looking at X-linked recessive phenotypes. Remember, this is specifically for recessive traits that are found on the X chromosome. In this case, they're going to appear much more frequently in males than in females because those males only have one X. They don't have an extra X to potentially cover up the recessive allele. Another thing to take note of is if there is a male carrying the mutation, he can only pass it on to his daughters. His sons, on the other hand, are going to get the Y from him because they can't get the Y from the mom. Now, daughters in women who receive one X-linked mutation that's recessive, but also carry a dominant X, are called heterozygous carriers. And it's possible for this mutant phenotype to skip a generation. Now, another thing to consider when you're looking at genes and traits that are being passed on is to consider mitochondrial and plastid DNA. Okay, remember, the mitochondria has its own set of DNA. Now, because this DNA is found in the mitochondria and not in the nucleus, it doesn't recombine when sex cells are fused together. The mitochondrial DNA is going to come specifically from the egg cells that have the cytoplasm in order to create the new zygote. So, mitochondrial DNA can only be inherited from the mother. All of the organelles and genes that are found outside of the nucleus come from the egg, which is coming from the mother. So we call these maternal or cytoplasmic inheritance. Now, some of these organelle genes, like the mitochondrial DNA, or even in other organisms like plants, where they would inherit the cytoplasmic DNA, some of those organelle genes are important for organelle assembly and function. So the mutations could potentially have large effects. Now, when we're talking about prokaryotes, these reproduce asexually, which ends up giving genetically identical progeny or offspring. But they can also exchange genetic material with those around it. Transfer of genes from one individual to another without sexual reproduction is called horizontal or lateral gene transfer. This can increase genetic diversity along with random mutation. Now, the process of this lateral gene transfer is called bacterial conjugation. This occurs when a projection or a part that sticks out, called the sex pilus, initiates contact between the cells. Then the genetic material is going to be passed from the donor to the recipient through a conjugation tube. Now, when this happens, there's no reciprocal transfer of DNA. The DNA is just going in one direction. The recipient cell that got the donor DNA is going to use that DNA and allow crossing over, and it's going to be then incorporated into the recipient's genome and therefore passed on to its progeny. A lot of bacteria also have things called plasmids. These are small circular DNA molecules that replicate independently from their main genome. Okay, these plasmids often have genes for unusual metabolic tasks, such as like breaking down hydrocarbons, or genes for antibiotic resistance. These plasmids can move between cells during conjugation. Here's a little example of how this can occur.